All right, so just a reminder, we are 1 week out from our next exam. So, seeing I am muted still. Okay. I didn't figure out what happened last time, obviously. Okay, all right, well, if you can hear us at home, then we'll just keep on going. So, good. All right, so yeah, obviously we had the fail at the end of last time. So just to pick up, I want to pick up right where we left off last time. We only lost about 5 minutes. We spent most of last time going over, we kind of, the time before got kind of all, this is what a virus is. We talked a lot about viral replication last time. So remember, viruses are either temperate or virulent. If they're virulent, they always only do the lytic viral replication. If they're temperate, they can also do that lysogenic. And that lysogeny of bacteriophage replication, if we have lysogeny in viruses that infect animal cells, we call that latency. So we talked about some differences there, some terminology about, when the virus is incorporated into the chromosome, we either call that a prophage or a provirus. So just pay attention to that terminology. And where we ended last time, we were thinking about, well, how do you grow up viruses if you want to study them in the lab? Of course, we can grow them in animals, and sometimes that's kind of the best way to do it. It's the most expensive way. We can grow viruses in eggs. We can grow them in bacteria. But remember, we can't grow viruses just on media. They have to have a living cell to reproduce in. So we're not going to see viruses growing up on nutrient agar. Another option is to grow them in some kind of eukaryotic cell. And that's where we left off last time. We were just talking about how this is less expensive than growing viruses in an egg or in an animal. You can use diploid cells, which are really embryonic stem cells. They grow for about 100 generations before they die, or continuous cultures where you use cancer cells, like Hela cells. And they just kind of grow on and on and on forever. Okay, there are some problems with using Hela cells. They have changed over time. So, it's kind of hard to have a standardized, cell line when they've actually evolved a little bit. But okay, so that brings us kind of to the end of our discussion. Now that you know all about viruses, thinking about that question, well, do you consider viruses alive? And that will continue to be a topic of debate. But in general, if you look at what we consider, the things that an organism would have to fulfill in order to be considered alive, organisms always grow. Do viruses grow? Generally, we would say no. Like, you don't get baby viruses that grow up into adult viruses. They're just assembled. Do they reproduce? Yes, but they have to be inside a living cell. They're very good at that, but again, they have to be inside a cell. There are some bacteria that have to be inside a cell to reproduce as well. Are they responsive? So what does that mean? Do they respond to stimuli? Do they change their behavior? This is the point that's a little bit more arguable, because yes, viruses evolve. We know that they may have a very high mutation rate. And so they certainly evolve. Do you consider that responding to a stimuli? A lot of people would say no, but some people would argue yes. They can evade host immunity. You might consider that responsiveness. So that's a little bit more up in the air. Do they have metabolism? Definitely no. Okay, they get everything that they need from their host cell. They use all the host cell's energy. They don't have to produce any of their own energy. So most people would argue no, that they don't fulfill enough of these things to be considered alive, but still people like to argue about stuff. I just want to take a few minutes to talk about prions. This is at the end of chapter 13. They also spend a little time in your book talking about something called viroids, which are kind of little messenger RNA particles that infect plants. We're not going to worry anymore about those, but prions are really quite an interesting thing. We knew for quite a long time there was some sort of agent that wasn't a virus or wasn't a bacteria that was causing disease. And in 1982, Stanley Prusiner coined this term prions. We're starting to understand a little bit more about what these particles were. Prions they're proteinaceous infective particles. So we pulled the name prions from that. So what's really interesting about prions, and it's correct to pronounce it prions as well, it kind of depends who you learn from and where you live. What's interesting about these is that there is no nucleic acid. There's no DNA, there's no RNA in these particles. So that begs the question, how do they reproduce themselves? That question, we're a little bit closer to understanding. We know that prions are a misfolded protein. So we all have this protein in our bodies. This stands for prion PRP. Sorry, stands for prion protein. Sorry about that. Okay, and we abbreviate that. So PR, prion, P for protein. So we all have this prion protein in all the cells of our body. It's a protein that's associated with our cell membrane. And we don't really know the function of it, but we know that we have it. We do know that if that protein becomes misfolded, then we call it prion PRP. So in your figure in your book here, the normal PRP is they refer to as cellular PRP. And then when that becomes misfolded, they call it prion PRP. Okay. So we know that exposure to that misfolded prion protein, causes normal PRP to misfold. Okay, so how does a prion protein make more prion proteins? Basically, it just exposes itself to the normal proteins. So, one bad apple spoils the bunch. One misfolded PRP influences others to misfold somehow. And I couldn't tell you the mechanism for that, but if you look at the structure here for the normal PRP and then look at the misfolded one, these green lines, those refer to beta sheets that you see. You see alpha helices in both. The beta sheets you see in the misfolded PRP and not the normal PRP. Okay, so that misfolding causes that protein to kind of glom up together. Neurons stop functioning, and eventually it impacts the just neural processing of the organism, and you see these big plaques in brain tissue that shouldn't be there. Okay, so when you think about, well, what are some examples of prion disease and how do organisms contract it? Well, understand that all these prion diseases are very rare, thankfully. And we'll look at some examples here in both animals and humans. All right, so probably the best known prion disease is the bovine spongiform encephalopathies. This is also known as mad cow. Okay, so that really came to the forefront in kind of the late 80s. Kind of the epicenter was England. For some reason, it had been common practice for hundreds of years to feed animals, ground up parts of animals that we weren't using for anything else. So bone meal, parts of brain were thrown into animal feed. And so when a cow died of mad cow disease, it would get ground up and fed to other animals, and that's how it was spreading until we figured out that we just couldn't do that anymore. So cows with mad cow would kind of stumble around. Again, they'd have these plaques in their brains that just was causing them to lose neural function. And I was in England for several weeks in the early 90s, and they kept telling our group, don't eat beef, don't eat anything with beef. And it was really, I didn't really understand, because I thought, well, we cook it, so why wouldn't it be okay to eat? Well, to deactivate a prion, you have to cook way hotter than cooking temperatures. You have to bring the temperature up to like 400 degrees Celsius for like 4 hours. So normal cooking doesn't deactivate it. This variant Creutzfeldt-Jakob is what humans got when they ate contaminated meat. All right, so again, this is kind of the human form of mad cow. There's only been about 200 people contract this. And again, that was, I don't think there's been any recently. Creutzfeldt-Jakob, so regular CJD, as opposed to variant CJD, this is when you just have a genetic, it's passed on genetically. Okay. So the genetic form of mad cow, basically. Scrapie is prion disease in sheep. It causes them to act really oddly. They'll often scrape up against posts and wear off the wool and even wear down their skin. Chronic wasting disease is in deer, elk, and moose. And you hear about cases of this in Michigan, which is kind of worrying because, of course, people shoot and eat venison. There haven't been any cases of prion disease passed on to humans, at least as far as we know, from eating deer with chronic wasting disease, but we worry about that for sure. And there's a few other ones. There's a fatal familial insomnia that's again very rare, but they've documented has been passed on in families. So that's one prion disease that's inherited. There's another one, Kuru, which has now been eradicated. That was a tribe in Papua New Guinea that's part of their custom was to, when their family members die, to show them honor, they would eat them. So they would, if you were really honored, you got to eat part of your loved one's brain. And that's how that prion disease was getting passed. That's been eradicated because they've been taught not to do that anymore. So prion disease can be just inherited, again, very rare, sporadic. So there's not a mutation in the gene, there's just a kind of rare misfolding of prion, that PRP protein, or infectious. So you get it by ingesting some animal that's been infected, or we think deer, elk, and moose can give chronic wasting disease to each other just by, sharing bodily fluids or just really close contact with each other. We're not totally sure about that. Right now, we don't have any treatment for any of these diseases. They're all fatal. There are some treatments in the works. There's some drugs that try to kind of jumpstart our mechanisms that we have to correctly fold proteins that have been misfolded, but eventually it can't keep up with that. So there's some therapies kind of in the pipeline, but nothing been approved yet. We're kind of still in the process of testing that on animals. Okay, Questions, anything about viruses, prions, anything? Okay, so we will get to some more specific viral diseases closer to the end of our time this semester. Okay, so we're kind of switching gears here a little bit. You have a lot of basic information now about microbes. We kind of focused on bacteria, but we talked about other microbes like viruses, protozoa, fungi. We talked about their metabolism, how they reproduce, how we see them under the microscope, et cetera, et cetera. We're going to think for a while now about how we're trying to kill those microbes. All right, so chapter 9 is focusing on microbial control. So I like this cartoon here, bacteria free, just means that I'm not going to charge you for it. All right, so we're going to explore ways to kill bacteria in chapter 9. Chapter 10 is all about killing microbes, bacteria in our bodies with the use of antibiotics. We'll be talking more about the process of infection and then get into a huge section on our immune system, again, how we're killing microbes, and finish the semester with, again, some kind of what we call our bug parade. So specific organisms and the infections they cause. So historically, right, people haven't known, until about 150 years ago, people didn't really understand what caused disease. So they had a lot of superstitions about how to keep themselves from getting sick. This was personal protective equipment in the 14th century. So doctors wore these gowns when they were going out and trying to help people that had bubonic plague, especially. They had they put on these beaks. They would stuff herbs inside those beaks so that they wouldn't have to smell the smell of death, because their best guess was that disease was spread by bad air. So they wanted to keep themselves from breathing in that bad air as much as they could. We didn't know how to prevent disease. There was all sorts of, crazy things that people would do. They thought smoking would keep away disease. So you would see even little kids smoking pipes. Bathing in urine was popular for a while, apparently. Tying a dead chicken to your wound, not a dead chicken, a live chicken that had been plucked. That doesn't sound fun to me. Leeches, not bleaches. Leeches were very popular for a long time because they thought that you needed to balance out the fluids in your body or get rid of the bad blood. And so, of course, what it mostly did was weaken the patient rather than helping them. So we are going to explore both some physical and chemical ways to control microbial growth. So we need to talk a little bit about some terminology and kind of go through a list of organisms and think about which are the hardest ones to kill and why they are. We're going to talk about just factors that we have to think about when we're choosing a method to try to cut down on microbes. We're going to spend some time on methods using heat. That's a very effective way to control microbial growth. We're going to talk less about chemicals, so I'm not really going to talk about this again. I forgot to cross that out. We'll be talking in general about the positives and negatives of different microbial methods. The few chemicals that we are going to talk about, bleach is one of them. So why do we put those in opaque containers? And of course, we got to talk a little bit about hand washing. That's a pretty important thing in the middle of a pandemic. What's the best way to wash your hands? All right, so I used to write out a bunch of definitions, and then I realized in your textbook, they had everything defined there pretty nicely. So I'm going to refer to that. It's got some examples here. It's a pretty nice table here, table 9.1. highlighting a few things. So these are terms that you've probably heard before, but we're going to make sure we try to use them correctly. So the difference between an antisepsis and a disinfection. So an antiseptic and a disinfectant. You might have used those terms interchangeably, but if we were to use them correctly, an antiseptic is an antimicrobial that you're going to use on a living surface. All right, so soap is a good example of an antiseptic. A disinfectant then is an antimicrobial that's designed to be used on some non-living surface. All right, so the alcohol that you spray on your bench in lab, that's meant to be used as a disinfectant. Disinfectants are formulated not to worry about whether they're harming somebody's skin or not. Okay, so antiseptics are typically a little bit less concentrated, safe to be using on your skin. Okay, you know, we could get our hands in bleach and that's not going to harm us, but you wouldn't want to do that over and over again, day in and day out. It would be very irritating to your skin and might cause other problems. These terms side and sidal, they're not terms, sorry, they're suffixes, or stasis or static. If a chemical is termed cidal, say it's bacterial cidal, that means that it kills bacteria. Fungicidal, a fungicide is one that kills fungi. If something's considered a stasis, it's bacterial or bacterial static, for instance, that just means that it keeps bacteria from growing. So most of the time we're aiming to kill whatever is on that surface, but sometimes it's good enough to just keep it from growing. All right. Degerming, that just refers to, it's kind of a catch-all, but usually we're referring to some kind of mechanical method, so some kind of scrubbing in order to remove germs. When you wash your hands, some of that scrubbing action is going to be important to get rid of as many microbes as you can. And then the difference between these three terms, pasteurization, sanitation, sterilization. Pasteurization is actually a kind of sanitation. It's a reducing number of microbes. We're going to talk about that in a little bit more depth. Sanitation, again, is removal of pathogens. We're reducing the number of pathogens, typically to meet some kind of public health standard. So what's the difference between a disinfection and a sanitation? They're pretty similar terms, but usually when we use the word sanitation, again, we're referring to, you know, in a hospital setting or maybe a food service setting where you have to meet certain standards because lots of people will be affected by it. And if something's a sterilization, it kills everything, even endospores. So in practice, it's really kind of difficult to achieve that sterilization, especially by chemical means. Most often we're going to use heat when we have to be really sure that something's sterile, or most commonly we would. But there are some things that we need to be sterile that are heat sensitive. So we have other things as well. All right, so we'd like to think about how these actions are actually killing microbes and two basic mechanisms. Sometimes a antimicrobial method will kind of do everything, and sometimes we don't exactly know. But the two most common is that either the cell walls and cell membranes are going to be altered. Okay. And thinking about why it's important for a cell to have an intact cell wall and an intact cell membrane. We're going to talk much more about weakening the cell wall when we get to antibiotics, but if that bacterial cell, if that cell wall is not intact, it's going to be not protecting the cell like it normally would, and it's especially going to be susceptible to Think about if it's in a hypotonic medium, which normally it would be, water is going to be rushing into the cell, and that cell is likely to lyse because the cell wall is not intact to keep it from lysing. If the cell membrane if we lose the integrity of the cell membrane, things pass through the membrane that normally wouldn't. Remember, when the cell's trying to produce energy, it's very important to keep that proton gradient, have to keep the protons outside the cell. If they're diffusing in wherever they want, then it's not going to get energy from that oxidative phosphorylation in electron transport chains. We only want the protons to come back in through that ATP synthetase. And of course, if envelope viruses lose their membrane, then they can't attach to the appropriate host cell, and they're not going to be able to enter that cell and reproduce. Okay, the other way that antimicrobials work is by damaging proteins and nucleic acids. Okay, so if the shape of a protein is altered, that's not going to function properly. An antimicrobial compound could, just break some bonds, and that might be enough to kill the cell. And of course, we talked about last time how mutations can alter nucleic acid, and we'll look at that a little bit more as well. So when you're trying to decide what's the best way to kill microbes on some surface, there's a number of things that we have to keep in mind. So what site is it? So the site to be treated. Okay, is this a site where it's okay to leave a little bit of residue on it? What if it gets stained? Is that a problem? Is it living? Is it non-living? All things to think about. Most antimicrobial methods need at least some exposure time. Okay, even chemicals, like you buy cleaners at the store, and if you read the fine print, it says spray it on, leave it on for 30 seconds, and then wipe it off. Okay, they need at least a little bit of an exposure time. How feasible is that? What concentration of the chemical do you need to use? Usually, if something's more concentrated, it usually works better, but there are some exceptions to that. The more dilute it is, the cheaper it is. That's always a nice thing. Are the cells that you're trying to kill actively growing? We'll talk about this, especially with antibiotic use. When they're actively growing, they're most susceptible. They're easier to kill when they're actively growing. Okay, so the faster they grow, the faster they die. Sometimes temperature can play a role. You'll see this graph here, figure 9.3. I can't remember. I think it's applying some chemical to some surface. I forget which chemical that it was using, but it tested the killing power, or how long it took to kill, both at 45 degrees Celsius, which that's pretty warm, right? Body temperature is 37, so it's pretty warm if you think about in terms of Fahrenheit. It took maybe 4 minutes to kill almost everything. At room temperature, the killing power, it took a lot longer. So it takes longer, the molecules aren't moving as fast. Sometimes pH will influence chemicals in particular. If you've ever taken care of a swimming pool, you know that you have to carefully pH the swimming pool when you add the chlorine, because as pH rises, and I don't know, this might happen after a rain, something like that. As pH rises, the chlorine will start to dissociate. And so you don't want the pH to be like 8, because the chlorine's not going to work. You got to keep the pH maybe 6.5 or whatever. I don't know what the optimal is, but from that graph that would appear so. And lastly, one that we tend to kind of forget about is there organic matter present? So what I mean by organic matter, things, bodily fluids, feces, blood, dirt to some extent. When there's a lot of organic, when you have a mess to clean up, like a diarrhea mess, the tendency is, oh, just throw some bleach on it and then wipe it up. Well, that most of our chemicals don't penetrate very well through organics. So really what you want to do is clean it up first. Well, maybe put bleach on it to protect yourself, but clean it up first, and then you gotta disinfect it again. Because again, those disinfectants wouldn't penetrate the first time. Okay. All right, so those are kind of the things that we have to kind of think about. Also think about what organisms are you trying to kill? When you look at this list of organisms, hopefully you kind of think, wow, I know what all these things are now. Like we actually have covered a lot of material so far. This is just sort of a general idea of the organisms that are easiest to kill and the ones that are the hardest to kill. So not necessarily for you to memorize, but it's kind of a general idea. There's some exceptions here and there. In general, prions are very difficult to kill. Like I mentioned, you have to heat them very high temperatures for long periods of time. There is a chemical that's supposed to kill or deactivate prions, but again, you have to soak the, say, medical equipment in it for like 10 hours. It takes a long time. So they're hard to kill. Most of the time, though, think about it, if you're cleaning at home, you don't need to be killing prions. Like they're pretty rare. How would they have got there? You know, unless, you know, you had some weird circumstance. Even, you know, at home, do we need to be killing endospores? Probably not. If a chemical is able to kill endospores, we want to save that to be used in the hospital when you're cleaning up after a patient with C. diff, maybe. So we don't really need that level of clean. If you're trying to, you know, clean to that level all the time, then we worry that our disinfectants are eventually going to lose their potency. That we've already seen some organisms start to be resistant to some of our what we consider lower level antimicrobials. So do we want to kill envelope viruses? Yes. Okay, remember those are pretty wimpy in the environment. That envelope degrades really quickly. HIV is an envelope virus. You can't get HIV from sitting on a toilet seat, right? They just degrade really quickly in the environment. Remember, coronavirus is enveloped as well. So non-envelope viruses are way up here. What's the difference between a small non-envelope virus and a large non-envelope virus? I'm surprised there's that big a difference there. Maybe the smaller ones are just a little bit more stable. I'm not really sure there. The other sort of surprising thing to me here is that they put gram-positive bacteria here are really easy to kill. Gram-negative's a little harder. Apparently, disinfectants will penetrate really easily through that peptidoglycan. So I don't know. Certainly when those organisms are in the body, gram-negatives are a little harder to kill because of that membrane in our bodies, because again, things don't penetrate very well. But again, there's going to be some exceptions here as well. All right, so even within different kinds of protozoan cysts, there's some variations. But again, those aren't as tough as an endospore of a bacteria. So again, don't memorize that. Hardest to kill, easiest to kill is reasonable. All right, so we are going to talk about these physical methods of control first. We'll go through those, especially spending time on heat and a little bit of time on radiation, kind of touching on everything in between there. And then I've picked a few chemical agents of control. We're not talking about all of them that the book talks about. I'm talking about less of them than in previous semesters. I dropped off the ones that just aren't used very much, usually because they're too toxic to us. So phenol, for example, that used to be the gold standard of disinfectants. You know, you compared everything else to phenol, but phenol is so irritating to humans, not only to skin, but inhaling it, that it's hardly ever used anymore. So I thought, well, I'm not going to talk about it either. So hopefully we'll get into some of those today as well. So first, thinking about the use of heat. It's usually kind of the first thing that you think of if you really want to make sure that a microorganism is dead. What kind of damage does it do? It does everything, okay? Permanently denatures proteins, destroys membranes, cell walls, disrupts nucleic acids, everything. All right, and typically in a normal semester, we would do a lab in 213 on ways to measure an organism's tolerance to heat. So I thought I'd talk about that a little bit in lecture because we don't do it in lab right now. One of the things that we try to measure is an organism's thermal death time. Okay, in other words, the TDT, thermal death time. Okay, what is that? That's the shortest time that kills an organism at a given temperature. So basically, this is a way to kind of try to quantitate how tolerant an organism is to heat. Another way we say that is how thermoduric an organism is. Most bacteria can tolerate at least kind of short exposures to heat, but you know, there is some differences there. So what we would do in lab is we would take a test tube with a little bit of media in it, inoculate an organism in there, and then we would put that organism in some water bath. Here I just said, say it's in a 65 degrees Celsius water bath. Warm, but not, well, that's pretty warm. I mean, it's what is that in Fahrenheit? Warmer than bath water. But remember, boiling is 100, so it's not anywhere near boiling. We put that test tube in that bath water. Actually, we take out an aliquot before we expose it to heat, just to make sure it's alive. So that's time 0. And then after, put it in the water bath. After 2 minutes, take out an aliquot, put it on an agar plate, put it back in the water bath. Every 2 minutes, take out an aliquot, and then we'll incubate that plate, see what grows up. Okay, so in these results that I made up, these first 2 organisms survived after 2 minutes, but not 4 minutes. So the thermal death time for E. coli and Staph aureus would be 4 minutes. Okay, let me just erase that I wrote that there, so that's not confusing. All right, so the shortest time that kills an organism. Now we could refine that and test it at 2.5 minutes and 3 minutes and 3.5. But you know, from what the data we have, we could say 4 minutes would be a pretty good approximation. But this third organism, Bacillus subtilis, is surviving even past 10 minutes. So the best we could say was the thermal death time was greater than 10 minutes for Bacillus. That's not surprising because remember what's special about Bacillus, it's a spore-forming organism. So the vegetative cells in the medium would be killed by the heat probably early on here. But if there were spores in there, they would still be viable and germinate even past that time. Yes, let's see. Sometimes, there it is. Thank you. Okay, so spore former is going to survive longer exposures to heat and be more thermoduric. A little different way to test heat tolerance is by the thermal death point. This is a similar measurement. We're just kind of switching the variables around here. We're testing the lowest temperature that kills at a given time. So instead of lowest time at a given temperature, it's lowest temperature at a given time. So usually we would designate the time as at 10 minutes. This is a little bit trickier to ascertain in the lab because you have to have lots of different water baths at different times. But again, we can see some variation here. The thermal death point for E. coli then would be 60 because it's not surviving at 60. It would be 65 for Staph aureus. Bacillus survives even pretty hot temperatures, again, just because if there's spores in there, they'll germinate. Okay. All right. So what kind of ways do we use heat? What are some applications? So probably the first thing that you think of if you're going to use heat is to just throw something in boiling water. All right, so baby's pacifier falls on the floor, the dog chews on it. Okay, let's throw it in some boiling water for a while. That's a pretty good disinfection, or you could call that a sanitation. But it's not considered a sterilization because it takes about 30 minutes to kill endospores. Okay, so it will kill vegetative cells within 10 minutes, but to be considered a sterilant, it has to kill something in sort of a reasonable amount of time, and that's usually considered about 10 minutes. So it's surprising to me that, vegetative cells would survive more than, or even an endospore would survive more than 10 minutes. But I've read that in enough places now that I believe that. So again, boiling, technically not considered a sterilant. Another moist heat application is pasteurization. So we consider this moist heat because typically if you're going to pasteurize, say, some milk or fruit juice, you pass it through some long tubing that's within some hot water. So water just comes up, penetrates a little bit better. So you pass it through narrow tubing within some hot water. This is considered also a sanitation. We just are going to bring the temperature up high enough to kill microbes that would either spoil the, say, milk or make us sick. All right, so if you look at the parameters of a pasteurization, we changed how we do this over the years. When we first started pasteurizing milk, this historical pasteurization, you can see 63. Remember, boiling's on 100, so that's not nearly high enough to be killing everything. Milk, when it's straight from the cow, is incredibly rich medium for bacteria. So it really quickly gets contaminated with bacteria. And there's all sorts of nasty things that grow in milk. The big one, the big organism that we want to kill is the mycobacterium that caused tuberculosis. So cows get a bovine tuberculosis that they can pass to humans. But there's lots of other stuff, E. coli, Listeria, brucellosis, like Salmonella. There's a kind of cholera, like all kinds of stuff that people used to routinely get ill with before we started pasteurizing milk. And of course, that name pasteurization, named for Louis Pasteur when he used it to control the spoilage of wine. Grape juice. So he would heat up the grape juice to kill the bacteria, add the yeast. The yeast would ferment the grape juice into wine. So historically, this was kind of a long process, 30 minutes, but when did we start pasteurizing? Right around 1900. It wasn't until 1947, Michigan was the first state to say that all milk that was sold had to be pasteurized. So you can imagine any kind of regulation kind of takes a long time to be accepted. Farmers didn't want to have to do it. It added to their costs. We see the same sorts of things today when regulations are imposed, but again, cut down on all sorts of illnesses, especially, babies that would drink cow's milks were really just susceptible to all sorts of things. Today, the most common way to pasteurize is this flash pasteurization. So we bring the temperature up a little bit higher and then really short, just 15 seconds, and then cool it back down again. And that's enough, again, to kill the pathogens, the disease-causing organisms. There might be some thermophiles that are alive in there, but we don't care about those. They're not going to make us sick. And to cut down on the numbers of organisms that would spoil the milk more quickly. So it gives milk a much longer shelf life. So less of it is going to be wasted when people don't buy it at the grocery store. Overseas, it's more common to do these other two types of pasteurizations, especially this ultra high temperature sterilization. So if we sterilize milk, we can actually store it at room temperature. So if you go to grocery stores in Europe, most of their milk will be, just at room temperature. And you would think that that would catch on here in the US, but there's a big drawback to that. Anybody know what that is? Have you ever had that Horizon milk, I think, in the little red jug? Apparently it tastes a lot different. So because of that, it just hasn't caught on in the US, and more costly to transport something when you have to refrigerate it. But probably not going to change anytime soon. All right, so pasteurization, whichever way that you do it, is going to cut down slightly on several of the vitamin content. I think there's a couple B vitamins that it cuts down on, but again, it makes it something very safe to drink. According to the CDC, so is it safe to drink milk that hasn't been pasteurized? According to the CDC, in the US, yes, it is. So, if you live on a farm or whatever and drink milk from the cow, that's generally okay, because we milk very hygienically now, and our cows now are very healthy, right? The farmers want their cows to be healthy because a healthy cow is going to give more milk. A healthy cow will give 8 gallons of milk a day. That just blows me away. That's a lot of milk. So cows are healthy, we're milking hygienically, very low risk to getting sick. But if you are immunocompromised or pregnant, you should not drink milk that hasn't been pasteurized. So, all right. Something that interests me, pasteurization. All right, a third way that we use heat to control microbial growth is using a autoclave. And again, if you're in lab, you've probably heard your lab professor talking about the autoclave this or autoclave that. The autoclave is a big double-walled structure. It kind of looks almost like a big dishwasher. What it is really is a big pressure cooker. Okay, it uses steam under pressure in order to sterilize. Right, so why do we use pressure? The principle here is that when you increase the pressure, the molecules need more heat to actually change into a gas. So when you increase the pressure, the boiling point goes higher. All right, so we can kind of superheat the water up to 121 degrees Celsius, so higher than boiling, because we've added an additional 15 pounds per square inch of pressure. Atmospheric pressure is about 15 psi. We've added another psi, another 15 psi. So increase the pressure, way increase the temperature. We just have to keep those parameters for 15 minutes, and that's enough to even kill endospores. It takes the machine a while to come up to pressure and come back down again, so it takes a little bit longer than that. But the autoclave's really quite flexible. We use it for all sorts of things: media, glassware, plastics. We can even put swabs in there. Of course, you can't get everything wet though, so they're some things like oils or powders, or obviously anything that's not heat resistant, you couldn't put it in there. But we use it for all sorts of stuff in the lab. If you're interested, this is just kind of some of the inner workings of the autoclave. Anything, a liquid like this, you would have to put it in a pool of water to make sure that the glass doesn't explode, you would have to cover that up with some foil or something. So there are some kind of rules to follow when you're autoclaving something. And we always put some autoclave tape on something before we put it in the autoclave. The tape looks like this on the top, where you really can't see the stripes very well. And then once that thing has come up to the correct, parameters, heat and everything, when it's done autoclaving, you should see the black stripes. So it not only reminds us what we have, what hasn't been autoclaved yet, but it tells us that the autoclave is working correctly. And if you want to be absolutely sure that your autoclave is working, then there's some biological indicators that we can put in there, but we're not going to worry about that. They do break down from time to time and kind of a pain in the neck. All right, so usually when using moist heat is preferable because moist heat penetrates much more quickly than dry heat, but sometimes you can't get something wet, so you have to rely on some method that doesn't involve water if you want to sterilize something. So you could use a hot air oven. Again, the drawback here is that it requires longer times and requires higher temperatures. But, like a hot air oven that is 160 degrees for 2 hours, that's enough to sterilize generally. This is not very hot. This is actually 350 degrees Fahrenheit. So, you could use your oven at home to disinfect, glassware or whatever. It just takes a little bit longer than if you had the steam and the pressure to do it. But again, useful for things that you can't get wet. Burning could be considered a dry heat application. Most of the time you can't burn something and have it still be good for what you want to use it for, but an exception there is when you're flaming your loop in lab to make sure that it's sterile. Make sure you get it red hot. If you don't get it red hot, you're just kind of waving it through the flame. That's not good enough. It's not getting it sterile. So make sure you're doing that thoroughly. Oftentimes we'll burn our garbage just to make sure that it's not got any living microbes in there as well. All right, so the opposite of heat. What about using cold to control microbial growth? Using cold is just going to slow bacterial growth. So both refrigeration and freezing would be considered bacteriostatic. Okay, it's not killing microbes generally, it's just keeping them from growing. So we use the refrigerator all the time in lab, incubate something overnight and then put those cultures in the refrigerator just so they don't overgrow till students come in again. Now there are some exceptions. Of course, psychrophiles grow best in cold temperatures, usually somewhere between freezing and room temperature. An extreme psychrophile might grow really well in a refrigerator temperature, 4 degrees Celsius. Here's an example of an organism that's considered a facultative psychrophile. So it doesn't grow best at cold temperatures, but it still grows pretty well. And that's Listeria Monocytogenes. That's an organism that typically is responsible for has in the past been responsible for some major foodborne illnesses in the US. I think the most recent one was cantaloupes about 10 years ago. There was some cantaloupes that were contaminated with Listeria. In 1998, there was a Listeria contamination at a Bilmar processing plant in Zeeland, Michigan. Like 20 people were killed then. So again, this is an organism that can cause problems just because it grows really well in the refrigerator. And something like lunch meat that you might keep in the refrigerator for a few weeks, this organism is growing. We often, if we want to store cells sort of indefinitely, we'll store them at minus 80 degrees Celsius, kind of long-term storage. And then when we thaw them out, not all of them are viable, but typically enough of them are viable to be able to start that culture again. So cold temperatures, good for storing cells, not necessarily killing them. Other methods, though, to control microbial growth anytime we dry something out, right? Bacterial cells need water for metabolism. So anytime they're dried out, they're not going to survive. So here's some women drying apricots to preserve them. So we think of a lot of foods that you might dry out so that you can, they're good to eat for a long period of time. Lyophilization, that's another way to think about is that's freeze drying. So it combines, you're going to really quick freeze something, like maybe dip it in liquid nitrogen to freeze it really quickly. And then you put it in a vacuum. So you get sublimation. Sublimation is when water turns from a solid to a gas and skips that liquid form. So you get that frozen water, you just kind of, suck it off with the vacuum. And you can store cells this way or food indefinitely because you've taken all the water out of them. Microbes can't grow. So astronaut ice cream, or there's lots of, food mixes that backpackers can get, and, so they're real light to carry. And then you just have to add water to eat them. Filtration is a nice method to cut down on growth or totally get rid of any microbes for things that are can't be heated. Okay, so used to sterilize heat sensitive material. So you put a membrane filter in here, say there's some liquid that you need to sterilize that can't be heated. You put a membrane there, and we can get membranes with pores even small enough to make sure viruses can't get through there. You usually have to suck that liquid through with some kind of vacuum pump if those pores are really small. And then your whatever's in your medium is going to be sterile again, or it's going to be sterile. So some examples of things that we might want to sterilize this way: vaccines, any kind of drug or antibiotic. Okay, there was a colossal fail about, I don't know if it's been 10 years ago now. You might have heard of it. There was a number of people that got fungal meningitis from a drug that was improperly sterilized. So there was a pharmacy in New England, I think it was in Massachusetts, that's, whatever, must not have been doing their quality control and checking to see that this was indeed sterile. And there was a lot of patients that were taking a steroid drug for back pain. So they were getting injections of this steroid drug, and it was contaminated with, I think it was aspergillus, some kind of fungus that, a lot of people died from that. So, all right, but, vaccines or a lot of drugs, you wouldn't be able to heat them to sterilize them. So this is generally a good method. Of course, another way to dehydrate cells is by osmotic pressure, right? So remember, if the solute concentration outside the cell is really high, that means the water concentration is low. Water would go from high concentration in the cell outside the cell. So the cells are going to dehydrate when they're in a hypertonic environment. So we actually preserve a lot of foods this way. Jam, for example, you add a lot of sugar to the fruit, not just because it tastes good, because it's going to draw the water out of any microbes that might be in there to spoil it. Same thing, you add salt to meat, not just because it tastes good, but it's going to not dehydrate. I mean, it does dehydrate the meat as well, but we're interested in dehydrating the organisms that would spoil it. Okay. All right, so our last physical method here is thinking about using radiation. So we talked last lecture about ionizing and non-ionizing forms of radiation and how those can cause mutations in a cell. So I want to think a little bit more about using what kind of applications, what do we use radiation for? So remember, ionizing radiation is going to create ions. So it's pulling electrons off of other molecules. And in the process, that's going to, we talked about how it can break even both strands of DNA, oxidizing covalent bonds, creates hydroxyl radicals. So remember, hydroxyl radicals are one of those toxic forms of oxygen. It can just cause a lot of cellular damage, and again, pulling electrons off of other molecules. So when you use radiation, this is not using any heat, right? These are just, remember, short wavelengths of light. We'll talk about the different kinds of ionizing radiation in a second. It acts pretty rapidly. So in other words, it kills cells fast. There's some difference how quickly it works depending on the wavelength. Generally is going to penetrate pretty well, at least much better than UV light or visible light. Okay, so 3 different kinds we'll look at here: X-rays, gamma rays, and we can make just beams with just straight electrons. That would be the shortest wavelength. Okay, so ignore the picture of Hulk just for the time being here. Forms of ionizing radiation. So we could produce an electron beam. Okay, and this is going to be the shortest wavelength. Okay, and produced by cathode ray machines. So it's pretty fast. It's going to kill cells just within a matter of seconds. Interestingly, it doesn't penetrate through cells very well, apparently. You would think the shortest wavelength would penetrate the best, but not necessarily, apparently. Still lots of uses just for electron beams, sterilizing spices, medical equipment, probably the most common applications there. But because it doesn't penetrate very well, it's not going to penetrate, through anything that's dirty. So it would be just kind of a final step, make sure it's as clean as we can get it. It's not going to penetrate through solid things. Probably the gamma rays are going to have the biggest application. Where do they come from? They're emitted by radioactive elements. Okay. And the most common application here is to hit foods. So a lot of fresh foods and vegetables, obviously we can't heat them, we heat them when we cook them, but when they're, in the supermarket, you rinse them off, or the growers rinse them off. That's pretty much all that they could do, other than hitting them with some gamma rays. So here's some strawberries here. This is from your book, that were both irradiated with gamma rays, soon after they were picked. Or sorry, this group was irradiated. This group over here was picked at the same time and was not irradiated. So you can see the spoilage that has taken place here because, the fungi and the microbes weren't killed. And any food that's been irradiated with gamma rays is supposed to have this seal on it, or this symbol on it. Used to be there was only a list of a certain number of foods that could be irradiated. That list is growing longer and longer. A few years ago, when we had a big foodborne outbreak with spinach, spinach was added to the list. All right, so again, we can't wash the spinach with soap. We can't heat it before we sell it as fresh spinach. Irradiation is really a good answer. It has been kind of slow to be accepted because people think, oh, it's radioactive. I'm not going to eat something that's radioactive. And I grew up in the era where you worried about that a lot because poor David Banner, caught in a blast of gamma radiation, and now, he turns into the Hulk. But that is, obviously, it doesn't leave any residue at all. Foods are not radioactive Like, it's just something people afraid of what they don't understand. So sometimes it changes the taste just slightly, and it's probably different for different foods. But that's probably the biggest drawback. I think there's one of the vitamins that they think it lessens the content of just a little bit. But when you think about the advantages of increased shelf life and so much less food being wasted, that it kind of outweighs that. Okay, so the gamma rays actually penetrate a little bit better than the electron beam, so that's why it has a little bit larger application. Okay, and there's other things besides food. You know, they can use that for medical equipment and other things as well, medical supplies. All right, X-rays don't have as much application. They do penetrate really well, but in terms of controlling microbial growth, apparently they act really slowly. So because it takes too long, it's just not as, and plus, you know, having big X-ray machines everywhere is just not feasible. So, all right. So the other kind of radiation that we talked about before was ultraviolet light. Of course, we called this non-ionizing radiation. That, if you remember, forms a specific kind of mutation in cells called thymine dimers. Pyrimidine dimers, but most often happens with thymine due to the structure of that base. And I think I actually put a little diagram in your notes. Remember, this is what a thymine dimer looks like: two adjacent thymines bonded to each other. So drawbacks to using UV light: it doesn't penetrate as well as the ionizing radiation, but still well enough. So it, you know, passes through air really well, through liquids somewhat, not through solids. But there's lots of application in terms of treating water and cleaning water that are in widespread use now. So it's kind of hard to see in this picture what's going on, but there are these lamps down here in the water flow. You have to regulate the water flow so it's not flowing too fast, give it a little time for the UV light to kind of penetrate through a lot of the water. And this is often used as a final stage in cleaning up our water, sewage treatment plants. Instead of dumping a lot of chemicals in there, we spend a little bit more on electricity costs, but it's kind of a better, it works better, and it's also a little bit cheaper way to clean up that water before we release it back out into the environment. Okay. Now, UV is harmful to humans, so we don't want to get, you know, in the way of a UV lamp. UV light can cause sunburn and types of skin cancer. So we don't want to look straight into a UV lamp. That's harmful to our eyes, but still does have a lot of uses. I think at Grand Valley, maybe in chemistry labs, they use UV light to hit the chemistry goggles just to clean them up a little bit. I've seen toothbrush holders where you can shine a little UV lights on your toothbrush to kind of disinfect it a little bit. Here's another application in a hospital room where they've wheeled the UV lamp into a patient's room. It looks like they've cleaned it up, and just as one other tool to use, maybe the patient had a antibiotic resistant infection. You're just going to try to clean everything as well as you can. You wheel it in there. You can see they even opened the drawers here. Turn it on, leave the room, turn it on remotely, come back in 15 minutes, you know, turn it off, come in, move it around to a little different spot so it hits, you know, different surfaces, you know, and again, turn it on again once you leave. Okay. All right, how are we doing here? Okay, so in terms of different chemicals that we can use, like I said, I'm just going to name a few, and I'm going to order them in order of low-level disinfectant and antiseptics, medium, and high level. So the high-level ones are going to work against some of the microbes higher on the list. It's only considered a high-level disinfectant if it will kill endospores. All right, so the medium ones are going to be a little bit better, you know, medium, low level ones mostly work on those easier to kill microbes. But again, sometimes that's all you need. So probably the first thing that you would think of if you were going to use some kind of substance, you know, to kill a microbe is soap. All right, so soap and detergents, they're considered low level. What's the difference? A soap is derived from natural products and a detergent is synthesized, in case you're interested in the difference there, but they both work the same way. They're surfactants. So it means they reduce the surface tension of liquids. Okay, so that means they're decreasing the attraction between the molecules. There is some mechanical action that has to be present when you're using a soap or detergent. So, you know, we caught my daughter when she was younger, you know, getting the soap on her hand and then just rinsing it off into the sink. Like, that's really not doing any good. You really have to scrub a little bit. You're releasing that surface tension, pulling the molecules apart and washing them away. So you got to scrub a little bit. They also work by disrupting the cell membranes and walls. So here's an example of a detergent. You can see they're actually amphipathic. That means they've got a polar end and a non-polar end. And so they basically insert into membranes. And we talked about how that can be deadly for a cell to have a bunch of holes poked in its membrane. So one detergent that you probably heard of is Quaternary Ammonia. If you work in a hospital, I think they still use quats somewhat. Again, these are low level, so it's not going to kill endospores or acid-fast organisms. And that we are seeing a little bit of resistance. There's been studies where they've been able to swab like bars of soap and found and streaked on a plate and found Pseudomonas growing up. I've tried that a couple times and haven't gotten anything to grow, but they know that they are seeing some resistance starting with this common hospital-acquired infection, Pseudomonas, to some Quaternary Ammonias. So that's why it's considered low level. All right, so your book talks about some other low level ones. I'm going to skip over those, and I'm pretty sure you shouldn't run into anything in your homework there. I think I read through that pretty well. The halogens are considered intermediate level. So we'll just kind of introduce this a little bit before I'll let you go. But they're on, if you picture the periodic table, the halogens on the right side, they almost have their outer shell filled. So they want to gain an electron in order to fill up their outer shell. So they're considered strong oxidizing agents. They want to grab electrons from other molecules and fill up their outer shell. Okay, so they're pretty reactive. Here's a list of some of the halogens. The fluorine, of course, we put minute quantities of fluorine in our water supply. It helps our teeth become harder so we don't have so much decay. Bromine, the only application I could think of, some people put bromine in their swimming pools. It's supposed to be a little less irritating than chlorine, but we're pretty much going to be thinking about some applications with chlorine and iodine. They're probably used a little bit more. This advantage kills within 30 minutes. 30 minutes seems like a really long time, so I'm not sure. Bleach is considered one of the fastest acting antimicrobials. So usually going to kill a lot quicker than 30 minutes. They will eventually kill endospores, but you got to leave them soaking on the surface or whatever for so long that it's not considered high level. They're still considered intermediate. Okay, so strong oxidizer, primarily acting on proteins in cells, but probably is going to do some other damage as well. So next lecture, we will finish up these few chemicals, and I'm going to include that on the exam for next week. So chapter 9 will be on exam 1. Probably should be able to finish that up in about 10 minutes next time, and then we'll move on to the antibiotics, which won't be on exam 2. Okay? And then, of course, next Wednesday, you get the day off. mental health day. So, all right, have a good day.